Hello everyone, my name is Marc Palmada Flores. I am from Girona, Catalonia, and I am a PhD student in the Comparative Genomics Lab in the Institute of Evolutionary Biology in Barcelona. I will be talking about the novo genome assembly of the critically endangered Montseny brook newt, Calotriton arnoldi. To start with, I will give you an overview of the talk. I will start talking about the habitat of the newt, then talking about why is it the most critically endangered amphibian in Europe. I will explain you some of the giant genome sizes of animals out there and explain a bit of the project of conservation Calotriton Montseny. Later on, I will be talking about the assembly process of this species and compare the resulting assembly to other caudata assemblies. I will finalize giving you an overview of the recent results using the ddRADseq data of this species, analyzing the population structure. So this newt is found in the Montseny Massif in northeastern Spain, Catalonia. It's endemic from there, and it lives in 8 mountain brooks in 2 different geographic regions with morphologically different individuals, as you can see in the west and in the east. These individuals have a semi-aquatic life in a riparian habitat. The most critically endangered amphibian in Europe now is considered the Montseny brook newt. It is due to the little amount of mature individuals and the decreasing population trends for multiple threats. These threats are anthropocentric and are some like water management or habitat alteration. There are many genome sizes in the animal world, and newts have one of the largest, as you can see in the graph. So far, we have only 2 different caudata assemblies available, the axolotl and the Iberian rift newt. For this species, due to its threats, there was a project called Calotriton Montseny starting in 2016 that aimed to protect the species, their habitat, and learn more about the biology and ecological requirements as well as the threats it faces. So far, many actions have been done to conserve this species, including establishment of a legal coverage, habitat improvements, and expansion of 3 main breeding centers, which have ex situ breeding programs. These centers have also done some reintroductions and monitoring of the species. To better understand the biology of this species and inform the breeding centers to improve the reintroduction actions, we have decided to work on creating a new genome reference assembly for this species. We have worked inside of the Catalan initiative for the Earth Biogenome project that aims to sequence all Catalan species, and with the collaboration and support of the OR1 pilot project of Oxford Nanopore Technologies. To start with, we estimated the genome size of the species using Illumina data with genome scope. And we expect a genome size of 28 gigabases, a highly homozygous genome with a highly repetitive content. As the genome is expected to be huge, we need a lot of data. As we needed to extract a lot of DNA, we used 2 different individuals, and we decided them to be brothers. We have generated more than 550 gigabases of data, both for Illumina and Nanopore. And to be able to run the assembly process, we used a node with 48 CPUs with a memory requirement of more than 1 terabyte. The total running time to be able to do the assembly, the polishing, the purging, and the previous filtering has been more than 3 months and 13 days. And it is normal because we are working with a species that is 7 times greater than a human genome. The resulting assembly, as you can see in this snake plot, is has a lot of contigs. It has 47,000, it consists in 47,000 contigs. It has 23 gigabases long and has a contig N50 of 1.6 megabases. The completeness of this assembly has been very good. We can see in the Buscos results that more than 85% of the Buscos are a single copy and complete. And it's only missing 4.4% of the Buscos. Compared to other available Kawata genome assemblies, the Montseny brook newt assembly has the longest contig N50 and also shows the lesser amount of contigs so far. Axolotl genome assembly shows a larger genome assembly size with less amount of scaffolds and bigger scaffolds N50. More assembly metrics of this genome have been assessed in order to see if we meet BGP standards. Based on the false duplication and different base accuracy metrics, we are within these standards except for the QB. This might be due to the fact that we used Illumina data of only one of the individuals used to create the genome assembly, and also that we used Nanopore data to generate it. At the end, with this assembly, we aim to know if the results of ddRADseq data that show us a highly differentiated population structure, and you can see that easterns and westerns are clustered together and separated from an outgroup. We will assess if with the whole genome we see the same pattern. To conclude, we have created a de novo genome reference assembly for the most critically endangered amphibian in Europe, the Montseny brook newt, Calotriton arnoldi. The resulting assembly quality metrics meet most of the BGP standards. We have presented the most contiguous genome assembly at contig level so far for a Kawata species, and population analysis using ddRADseq data show high levels of the population differentiation that will be assessed. These results will be assessed with low coverage whole genome data mapped to the reported assembly. I want to thank a lot all the participants in rating the assembly that have been involved, and I want to thank you all for being here and for your attention.